So shall we start? So you're on mute.
Sure. Yeah, sorry. Whenever we are ready.
Good morning to all the participants. Welcome to the second session of today's FDP. With the kind consent of the convener, we are starting our second session of FDP. Our resource person for today is Dr. Raghuveer Ramachandra. He holds a PhD in Animal and Food Sciences from West Virginia University, Morgantown, USA, and he also holds a Masters of Science in Aquaculture from Indian Council of Agricultural Research. He is the CEO or founder of GaloreTx Pharmaceutical Private Limited. He's the founder of GaloreTx, a biotech startup focusing on discovery and development of novel therapeutics, small molecules for treating various indications including cancer and inflammation. He has led GaloreTx to be a mature revenue-generating small biotech, leading strategic and growth opportunities, providing overall leadership to GaloreTx to manage compliance, revenue stream, operations, business development, scientific and technical aspects. Before he starting his startup, he has also worked as a scientist in quite big companies, as a project lead in immuno-oncology and as a team leader in vivo pharmacology from 2008 to 2018 in a leading multinational company. He has worked as a senior scientist in Origin Discovery Technologies and also held quite a few key positions as pharmacology team leader, associate scientist, and also as a consulting scientist. He holds more than 24 publications in journals of national and international repute to his credit. He has also obtained quite a few fellowships from various organizations. Sir, on behalf of the staff, student, and the management, the Oxford College of Engineering, I, Salma, take it a privilege to welcome you to our today's session. So welcome to the session, sir, and you may kindly start the session.
Sure. Thank you, Sarma. That was a very kind introduction. I really appreciate Oxford College of Engineering taking this kind of faculty development programs, which are very important during the course of teachers and faculty's career. I think this brings us up to date with respect to what is happening outside of academic world, and it gives us also an opportunity to interact with you and see how we can cater to your needs and how we can address some of the concerns that you may have, and also how we can influence each other in terms of developing manpower, which will eventually either go to industry or academics. So I really appreciate this opportunity to interact with you all today. My email address and my phone number is noted here. Organizers also have this information. Please feel free to contact me during or after this talk with respect to any questions or concerns or any issues that we may want to talk offline. So I just want to, instead of just giving some gyan or just talking about some very scientific aspect, I thought we'll make it a case study today. How one of our programs was ideated, how we set up the process to initiate and progress this program, and what are the challenges and what are the issues that we faced, and how we took it forward all the way, and what is the current stage right now. So I thought we will make it that way so that it is interactive and it is more useful for you, if any. And feel free to stop me if there are any questions. I can stop and we can move forward after addressing those concerns. So agenda today is, I just want to start from a scientific hypothesis, because this is where academic faculty would be very familiar with. We all is proving or disproving them, and eventually how that can be translated into a product. in a company setting, in a business setting. How do we, what are the actionable items? How do we work on a business plan, and what are the unfair advantages that we may have as a team that other companies, other teams may not have? What are the flow chart that we chalk out and work on, and what is the business model? How do we eventually out license or cash out with this asset? What are the unique seller propositions, market opportunities, etc., and what are the complaints and other challenges that we may face, and what are the opportunities to start a science-driven business? We'll briefly touch upon that as well. So let's just jump in. So as you can see here, this is a purely highly scientific slide. I just want to start from here. Because this is how a science-driven organization will start its project, right? So we all keep reading current literature. We base all of our project ideas based on current literature. So such ideas are plenty, but we have to choose which one makes more sense in terms of current market conditions as well as unmet medical need. So on the right-hand side, the figure. This is basically a representation of cancer immunotherapy, how tumor cells interact with our immune system, and how in a healthy system, how the cancer cells are being chewed up or eaten up by our immune system. So if you start from one here, how the cancer cell antigens interact with cancer cell antigens interact with immune system. They are presented in the second stage. Cancer antigens are presented to the immune system, and then priming and activation happens in the lymph node, and then T cells are trafficked into the tumor through the blood vessels. And then infiltration happens into the tumor. They cross the barrier and then get into the tumor, and then they recognize the cancer cells, T cells, and then killing happens through various mechanisms. So outside of this figure, the list of items on the outside of this figure is how we tackle each of these stages, how we can exploit each of these stages to impart a therapeutic intervention. That is where our interest lies, right? So there are various stages where you can impart therapeutic intervention. Some of the things that are very recent are using cancer cells ability to evade from the immune system by expressing some of these proteins such as PD-1, PD-L1, and there are CTLA-4, and there are multiple other proteins. These are called checkpoints. So these are immune checkpoints. So when they are expressed, they are also expressed by our healthy tissues. When they are expressed, T cells recognize these tissues as healthy tissues and will not kill them. So cancer cells also express these proteins, which makes them avoid the killing by the immune cells, recognition by the immune system, and evade the consequences of being recognized by the immune system. So, so when these proteins are present, immune system will not affect them, will not kill them. So what we are trying to now, the state-of-the-art cancer therapies exploit this mechanism, and they inhibit these proteins so that cancer cells become visible to the immune system again, and they do the natural process of killing these cells by various mechanisms. There are CD4 positive cytotoxic effects, CD4 mediated, CD8 mediated, and then there are natural killer cells, and there are dendritic cells. There are various mechanisms by which cancer cells are killed and absorbed by the immune system. So, so the process, the beginning of that is to neutralize these proteins, PD-L1 and PD-1 kind of immune checkpoint proteins, to make them visible. So it is not a silver bullet, right? There is usually in the science, in our understanding, there is never a silver bullet. There is no one protein or one gene or one marker that can completely inhibit the tumor growth or completely kill the tumor cells. So there are exceptions for that rule, of course, but in this case there are multiple immune checkpoints like PD-1, there is CTLA-4, there is LAG-3, there is TIM-3. There are many other proteins that are being recognized in the literature now. So a go-to strategy is to combine these checkpoint therapies, for example, CTLA-4 and PD-1. So if you can combine therapies against CTLA-4 and PD-1, the efficacy is much more visible, much more effective. So therapies against CTLA-4 and PD-1 are approved for patients for multiple years now, many years now, I think 3-4 years now. What we have observed is therapies are very effective. If the patient responds to these therapies, they live a very healthy, disease-free life for many, many years. I think one of the first clinical trial with one of these antibodies is ongoing for more than 8 or 9 years now, and many patients from the first clinical trial are still alive and they're doing really well. So disease-free, they make patients disease-free. It's sort of a cure almost, we can call it. But the problem is not all the patients respond to this kind of therapies. 10 to 15%, 10 to 20% of the patients only respond to this kind of therapies. So obviously the next logical conclusion is to combine some of these therapies. So if 20% of the patients respond to CTLA-4 antibody alone and another 20% of the patients respond to PD-1 antibodies alone, why don't we combine them and see whether, if not 40%, at least 30% or 35% of the people patients respond. So that is the next step, and that has also been carried out. There are many clinical trials showing added advantage, added survival benefit when these kind of antibodies are combined. So now antibodies again come with a lot of disadvantages. They have to be administered as intravenous or intramuscular injections in the hospital setting, which adds to the cost of the treatment, and they have to be transported in a cold chain. And the production cost for monoclonal antibodies or recombinant proteins is very high because it's not an industrial process. It has to be carried out using live cells and things like that. So it is not in a chemical reactor that can be produced. So a lot of cost disadvantages are there. So more than the toxicity of the drug, it is the financial toxicity that affects the patient. So especially patients in countries like ours, where affordability is a big concern, and this kind of antibodies are very much out of reach. For example, PD-1 antibody costs about $100,000 per year per patient. So that's about 70-75 lakhs per year per patient, which is totally outrageous in terms of affordability in countries like ours. So what we are trying to do is, we are trying to come up with a much cheaper, much affordable option. So what we do is, so these are all protein-protein interactions. PD-1 and its receptor is a protein-protein interaction. So if you look at the figure above, so PD-1 and PDL1 is interacting with each other here in this figure. And then we look at this interactive interface where these proteins actually interact, and look at these amino acid sequences. These are proteins, right? So they are made up of amino acids, and look at this amino acid sequence and design a peptide that can go and bind to this interface and disrupt this protein-protein interaction. So PD-1 cannot bind to its receptor, either PDL1 or PDL2. So the signaling cascade does not happen, and so the tumor cells become visible to the immune system. So this pharmacophore, or the peptide, is further modified into incorporating drug-like properties like solubility or stability or oral bioavailability or good PK parameter and many other drug-like properties, and carried out, screened in multiple in vitro cell-based, mammalian cell-based, or PBMC-based, or other assays, and taken eventually to a mouse disease model. And then a novel medicine molecule is identified for taking into clinical trial. So using this, this is a novel transformatory approach, because people usually take a library of 100,000 compounds, 200,000 compounds, or 50,000 compounds and screen in a binding assay to identify a molecule to take forward. So we don't do that. At GaloreTx, we design our molecules based on the peptides, amino acid sequence. So this makes us have an unfair advantage because we have fewer molecules to screen. So we can directly go to a functionally relevant assay, even in vivo assay, rather than a biophysical binding assay. So in terms of our advantages, what are the advantages of this approach, compared to monoclonal antibody that we earlier talked about? In terms of toxicity, for example, antibody remains in the body for 2 to 3 weeks. And if a patient develops an adverse event, adverse reaction to the antibody, it is really difficult for the doctors, the physicians to manage this adverse event side effect. Because it remains in the body for 2 to 3 weeks. So you have to treat them with antihistamines or anti-TNFs, and it is a complicated procedure to remove this antibody from the body. Whereas our molecule, it's going to be a short-acting molecule, a small molecule. You just stop dosing. Within 24 hours, it is cleared using through either urine or stool. So then the side effect or the adverse event that is caused by the drug is eliminated within 24 hours, compared to over 2 to 3 weeks in antibody. In terms of dosing, antibodies are large size, large molecules. They need to be dosed as IV or intramuscular, whereas ours, we can formulate it in a capsule or a tablet so that a patient can take it from the comfort of their home. So no hospitalization required here, which brings down the treatment cost. In terms of response, since these monoclonal antibodies are highly specific, very specific to one target, and since they are antibodies, it cannot be combined. Many targets cannot be treated at once. Whereas in our case, since it is an oral tablet, we can combine multiple targets. Multiple proteins, which increase the response rate. Obviously, in terms of production cost, since antibodies are produced in a very complex and expensive recombinant process, the cost of production is very high there, whereas our molecules can be produced in a very cost-effective synthetic industrial process, which brings down the cost by more than 20 to 25-fold cheaper. So using this process, we have this approach. We have in our previous company, we have identified multiple molecules. One such molecule is currently in phase 2 clinical trial, and it is showing really good efficacy in terms of response, in terms of overall response rate and disease-free survival response in multiple indications. And that is actually ongoing. That is with our previous employer. So the same approach, we are using it in multiple programs in our current company. So how do we do this? So we know that it works. We know that. Let me go back here 1 second. So we know that combining these two, three, any one of these combination, either CTLA4 Vista, CTLA4 PD1, or PD1 Vista, any of this combination combining is more effective than single agent. So, and also we have a technology, we have an approach that is established to identify a novel molecule at a very cost-effective and timely manner, which is very important for eventual success of the molecule. So how do we do that? So we have to establish a flow chart to screen these molecules, the molecules that we design and identify and synthesize. We first screen it in a binding assay, if possible, otherwise a reporter assay. And then parallelly we carry out whether it is soluble or not, whether it is stable in plasma or not, and then we take it to a functional assay. For example, this one of this program, we know that that target enables proliferation of particular epithelial cells. So we check whether our molecules can stop that proliferation or not, and then we eventually take it to animals, do a pharmacokinetic study. In the meantime, we also
Dear participants, looks like there is a technical glitch at the speaker's end. Please stay with us. He will reconnect with us.
Sorry, I apologize for that. My internet was not very stable. So you can hear me all here?
Yes, sir, we can. Yes, sir, we can
Okay,
hear
great.
you.
Okay, okay.
Your
So,
PPT, kindly share that, sir.
oh, it is still not shared. Okay. Okay. So we employ either of these two business models where we take the molecule all the way till preclinical development and license it. So this whole process will take 18 to 24 months. We have to fund this program by ourselves if this is the case, this model we employ. In the meantime, we have applied for multiple government support, multiple grants, and some of them we have received and some of them are in the process of evaluation. So here the risk is all ours, but eventually we will get a milestone-based payment. Whereas in model 2, we can collaborate with a partner with a biotech or a pharma company at various stages at any point of this time and in this course of drug discovery, and then receive research funding and milestone based funding during the course of this. So we basically co-developed this drug discovery program. So both of these models are being used right now. We have one program that is being supported by a collaborator, and 3 other programs that we are running on our own. They are at different stages now. We have not out-licensed any of these assets yet. So basically, this is how we design our business model so that we can be sustainable as a company, as a business unit. So what are the unique selling propositions for us? Basically, we are quicker. In a pharma company, I have worked in a pharma company for more than 10 years. I know how much time it takes. So I can tell you for this 18 to 24 months, usually it takes about 4 to 5 years in a big pharma company, because there are multiple checks and balances involved in a big company. And the decision making is a little slow because there are multiple meetings that need to be held, and certain procedures have to be followed. There are certain flow charts, and there are some different checks and balances that we don't have as a startup. We can be very nimble and very quick to take a decision, and our overhead cost is much more than an order of magnitude lower compared to a bigger company. So we are very cost effective and much quicker to identify a candidate. That is one of the most important USPs for us. And we, because of our approach, because ours being a small molecule, we have the convenience of oral dosing with our molecule. So that is a very important advantage for us. High tolerability because it is a peptide-based approach and it gets cleared within 24 hours, as well as these molecules are amino acid inspired. Because of that, it is highly tolerated, and of course significantly lower cost because of non-requirement of cold chain and industrial process of synthesis and so on and so forth. So in terms of market opportunities, $4 to $8 million is the upfront value for a preclinical asset during these years. So a candidate that can show preclinical efficacy and safety usually fetches about 4 to 8 million or so. But we have to fund this program until this stage. So that is basically where the challenge is. So in terms of competition, monoclonal antibodies are approved. Multiple of them are approved right now for use in the clinic in the patients. So combined, those monoclonal antibodies sell, make a lot of money for these pharma companies, Merck and J&J's and Novartis and so on. So we expect to replace part of that patient population. Of course, not us directly, but our partner who will take it to clinic and eventually market it will replace part of this market with our molecule. And then there are other agents that do not directly target these proteins, but target downstream pathway. They are not really considered as our competitors because they will have off-target liabilities, toxicities that are involved with the chemotype, where we don't have that kind of issues. So this is basically how we monetize, or how we take the molecule all the way till the market, how we out-license our assets. So I just want to also give you a brief glimpse of what we go through as a company. So how do we incorporate a company? We have this idea, we think we can make this happen, but how do we incorporate? What are the actual steps to incorporate a company is a different ball game. We have to decide on a name. Nowadays, Ministry of Corporate Affairs is very stringent on giving names also. So they will not give you a generic name or a dictionary word. You have to have a unique brand value, name, and then you have to incorporate with different articles of association and memorandum of association, which determines what activities are you going to do as a company. So those you have to very carefully draft these things, because that can affect how you do business, what kind of the business you eventually do. when you come into existence. And you can incorporate your company as a LLP or limited liability partnership or a limited liability company, LLC, or a private limited company, or even a proprietorship. But most of the government grants and compliance issues come up when we use proprietorship as a vehicle. Usually private limited is a preferred way, because external investment can be much more straightforward in that case. So we also have to comply with a lot of labor laws. PF comes into picture, and ESI comes into picture, and different types of working environment conditions, salary requirements, and all of those things are also need to be taken care, because now we are not dependent on a bigger company's HR department or a finance department. We are a company on our own, so we have to look at all of these things. Preferably have a consultant who can advise us on all of these things. GST, of course, has streamlined a lot of things. It has replaced many different types of taxes into one umbrella tax across the states. But it also comes with a compliance requirement. We have to file few returns every month, every quarter, every year. There are different types of issues associated with that. But applying for a GST and getting a GST number has become extremely simple now, just an online process which takes less than a week. And coming to bookkeeping, so what kind of purchases you made, so GST invoices, and what kind of investments you took, and how much money is spent as a cost of goods sold, or what is the income, what you sell, and all that. So this bookkeeping comes into the picture from day one, and also it is extremely important to observe good bookkeeping, because eventually when we out-license these molecules, every reagent, every cell line, every molecule's ingredients, the reagents and supplies have to be tracked back to an original authorized supplier, right? Because people don't want to buy an asset from you if things are done in a not so legal way. So this bookkeeping gives a track of each of these material flowing from authorized distributor to you, and you selling eventually to the in-licensee of your product. So very important to keep these things in mind from day one. If at all any of you have ideas of starting your own company, as I don't know if it is allowed in Oxford to start a company by a faculty, or at least advise a company started by students or incubated at your institution. All of these things are extremely important. It has to be kept in mind. Import-export code is another issue. We have to obviously we have to import many reagents, kits, and cell lines, and samples, and different other things from different countries like China or US or Europe or anywhere. You need to have something called as import-export code, without which we cannot import any of the reagents, any items. So this process also has become very simple, but customs and border protection is a very tricky area. You have to be very extremely up to date with what numbers you need to use, and what procedures we need to follow. And IBSC, since we are in biological research, we at some point we use recombinant proteins or cell lines or viral vectors and so on. That requires us to have compliance with the Institutional Biosafety Committee. This is administered by RCGM and DBT. I'm sure your college, your institution has an IBSC, but as a company, a smaller company, however small it is, you're supposed to have an IBSC. You have to report to RCGM and DBT on all the activities that you carry out with respect to recombinant DNA, mammalian cell culture, and viral vectors. So this is also, it is a pain in the wrong place, but these kind of checks and balances are there for a good reason, because there is always a fear of misutilization of these resources. So um, very important to comply with this, because something goes wrong, all of those issues will come gushing back at us. So Institutional Animal Ethics Committee, IAC, is another issue. This is not so straightforward. This is very complicated. It has to, it's a big topic by itself. But if you are using a contract research organization for doing your animal ethics committee, animal experiments, then you don't have to worry about this. But if you have your own animal house, then you have to have constitute your own animal ethics committee and follow those guidelines. CPCSCA determines all the guidelines for this. And then Pollution Control Board approvals are also absolutely required to carry out any type of industry, however small it is, however big it is. But if you are in an academic campus or an incubator, usually some of these issues are taken care by the incubator or the academic institution. So usually it is straightforward to rely on an incubator, so that we can minimize those issues, compliance issues. One of the most important part is to protect your IP. Of course, all you generate, do all of this work for 18 months, 24 months, all you generate is intellectual property. Structure of a molecule is all you generate. There is nothing else. There is nothing physical, basically. So that protecting that has to be of paramount importance. We do that either by filing provisional patent applications, or it could be even a trade secret or a trademark, other forms of protecting IP. But preferable way is to file a patent application, so that the rights for that patent can be transferred to another company when we are out licensing our molecule to a different company. So so some of these issues are they look overwhelming, but they are there for a reason. They have to be followed, and if we miss any of these things, it will come back biting us at some point of time. So they are all necessary evils, basically. To start a company, I thought I'll just list a few of the challenges that we faced and that are currently stopping many of the faculty or many of the researchers from starting a business. The most important, I think, is funding opportunity. Funding ecosystem has not evolved in our country, especially. If you have a great idea, in a Western country, people will come to you and give you a few $1 million and ask you to start this business and support you. But that kind of support or the ecosystem has not happened. That has not evolved here in this country. The investors would rather invest in a real estate property where they get a 10% return year on year, than invest in a biotech company which may take over 5 years to materialize. Although we say that there is a 40-fold, 50-fold, 100-fold return on investment, but they know what works in real estate or retail or a different type of investment. So they would rather prefer that. So that kind of ecosystem, that mentality with respect to the investors, high net worth individuals, or even institutional support has not evolved. Although it is a continuous process, there are people who are coming forward to fund this kind of companies, successful entrepreneurs, for example. But that has not evolved, but there is some support still. I have to mention here about government support. Both the Department of Biotechnology, Government of India, through BIRAC, and Karnataka government through vehicles like Elevate, Idea to POC, and other schemes. They have been supporting entrepreneurs, technology-based companies. We have also received multiple of these awards. We have received Elevate Karnataka and Big Biotech Ignition Grant, and there is also a funding mechanism called Nidhi Prayas. So that also we have received. So government support is very very much an emerging area, but also very much needed area. And it is not impossible to get this support. We don't have any influence, or we have not paid any bribe, but we have received that support. So that we have to appreciate that mechanism. It is evolving. Venture funding, as I told you, it is not really existent here, but still there are some small venture houses which keep looking for small biotech companies to fund. Institutional funding, like big banking structures or consortium of banks and financial institutions, this essentially doesn't exist in India. There are some angel networks which fund small startups, promising startups. So we have to explore all of these opportunities to see how we can fund our biotech company. So, so with respect to compliance, there have been a lot of effort from the government side, both central and state, towards simplifying the compliance requirements. One of the most important development in this regard is emergence of incubators, business incubators, technology incubators, TBIs is what they are called. Once you get into an incubator, quite a few significant compliance requirements go away. For example, pollution control board requirements, or building code and shops and establishment requirements, compliance requirements. They go away because incubators take care of them. You still have to have your own bookkeeping, your own income tax filing, GST filing, and import-export code, and GST, and so on and so forth. IBSC, for example, also. But still, pollution control board and shops and stuff. These are some of the bigger ones which take 6 months to 1 year to set up. So those go away. So incubators are very important, very helpful in terms of setting up a business to start hit the ground running. So that they have lowered barrier for entry to a great extent. So some of these incubators also have funding, which also support your program in terms of consumables or rent or paying salary and so on. So incubators have been really active, and they're playing a very important role in lowering the barrier for entry. There are some government initiatives like ease of doing business and single window clearance and some of those things. I would say that is still an emerging process, and we still need to go a long way before we say that we have achieved ease of doing business. But a lot of things have improved, I have to say. In terms of manpower, there used to be a long gap between academic training and the industrial requirement, job profiles. So schemes like BICEP and BCIL, BICEP by Government of Karnataka, BCIL from with the DBT support, they have been doing a really very good job of bridging that gap. So they put the students in companies for 6 months internship, including a pay for them. So that those students can get used to the business environment, what is required in the company setting, in an industrial setting, acquire those skills, and they will be ready for getting absorbed in the same company or apply for jobs in different companies using that skill set as a as an asset, as a learning. So I think these three challenges are the most important challenges that we face before we start a company, and some of them have improved significantly, and some of them have a long way to go. But I would say there is no better time to start a company if you have a good idea than now. So and any of you faculties, any of you have good idea that can be taken up as a business model, business plan, as a product, has a potential to be marketed, I think you should do it now, because things have significantly improved, and there is a hope that it will significantly improve in the near future. There are challenges, but there are also people who have overcome those challenges, so you can always consult with those people. And I'm doing the same thing. There is a small but very closed and very interactive ecosystem of startup folks in Bangalore. We interact with each other on a regular basis with respect to any help required, reagent required, or knowledge in any of the issues. So if any of you need any of those information, please feel free to reach out to me. I'll be happy to introduce you to relevant people. If I can help you, I'll be more than happy to help you. But I'm sure there are issues that I cannot help you with. I'll be able to put you in touch with folks who know, who have been there, done that. So I think with that, I conclude this talk. So if you have any questions, maybe next half an hour or so, we can discuss and conclude this session. I hope it was helpful for you.
Thank you, sir, for that informative talk. Participants, if you have any questions, queries, you may please unmute yourself and ask. Sir, do you provide any internship or project doing opportunities for our BTech students?
We do. We currently have 3 students, BTech and MSc chemistry and different streams. Yeah, right now we have already 3 people. Since ours is a small facility rented in an incubator, our space is limited, our resources are limited. Right now we cannot take any more, but yeah, there may be positions open, so we will be in touch. We will communicate with each other to make sure when there is an open opportunity, we will look at it.
Sir, is there a time where they are supposed to apply? Like, do you call for these internships? How is it going to happen?
No, usually it is a rolling kind of recruitment. When these interns they decide to move on, then we have to replace with somebody with them, with a new fresher, and there is no set window for recruitment, and also throughout the year somebody will leave and we have to replace them. So it's a rolling recruitment cycle. There is no set window. But do you have association with PCIL or BICEP?
No, sir. No, sir.
Okay,
Right now we don't. BCIL, we are in the process of MOU.
Okay. So no, if that is incorporated, you have arrangement with BCIL or BICEP, then you will have 2 cycles a year. So then you will have a cycle, but we will have a rolling requirements. Yeah.
Participants, any questions? So there are no more queries coming in from. Okay. So we
Okay.
have
Yeah.
one.
Thank you very much. Yeah. Tell me.
Sir, it is a very useful session. Those really interested to become an entrepreneur and startup. Thank you. So he's complimenting you, sir.
Thank you very much. I hope it was, you got some idea about what we go through and what are the steps we take. So hopefully, if any of you are interested, please reach out to me. I'll be happy to help.
Thank you, sir. So on behalf of the student, staff and management from the Oxford College of Engineering, I, Salma, extend a heartfelt gratitude towards Dr. Raghuveer for taking his time out from his busy schedule and being part of this FDP and sharing his achievements and his hardships and his journey towards making a startup. So thank you so much for being part of this FDP program.
Thank you. It was pleasure interacting with you. Thank you very much.